And as you said, my name is Annemieke Geluk. I work at the Department of Infectious Diseases, which is now merged with other departments to the Leiden University Center of Infectious Diseases, called LUCID. And and at that department, I work in a group that studies mycobacterial diseases. And the mycobacterial research group at LUCID focuses at both the pathogens, development of vaccines, development of drugs, and also diagnostics. My group mostly works on diagnostics using the immune system as a readout for that. But for this series of presentations, I will not go too much into detail into that. So as a way of introducing, so my group is focused on immunodiagnostics of mycobacterial diseases. And we are both based in the lab doing basic research, as well as translating that research to the field, to field situations. And as you can see here, so those two pillars are supported by my group members, most of whom are PhD students, and some postdocs on research technicians, and PhD students coming from all over the world. So from China, Brazil, Pakistan, and from also from several African places, including Ethiopia and Madagascar. And so this is some reflection, so you get an impression what we do in the lab on the left. So the lab can be in Leiden or it can be in any other part of the world where we perform research and try to also do capacity building, so to transfer that research, transfer that technology to those labs. And on the other hand, work in field situations, because of course the diseases on which we work, they occur mostly in tropical areas. And so we try to translate to the field, and therefore we also provide training in the field to the field staff and to the lab staff at these specific places. So first, a little bit about the pathogens that we study and the diseases they cause. So the main pathogenic mycobacteria are Mycobacterium tuberculosis, that causes tuberculosis, a lung disease, which is deadly and which is very contagious. And also next to COVID, the first killer disease of a single infectious agent. And so it can be treated with antibiotics, but that takes about 6 months. And there's a lot of resistance, so multi-drug resistance, resistance against the antibiotics used to treat the disease with. On the other hand, we have Mycobacterium leprae, and it's very similar Mycobacterium, but still very unique also. And it causes leprosy, and leprosy is a disease of nerves and skin. It is not deadly, although it can cause handicaps that are long-lasting. So and they will not be, and they're irreversible. However, the disease is not very contagious because you need long, intense contact with a multibacillary source in order to get infected. And but also the treatment is very long, so using 12 months of antibiotics usually. However, an advantage for leprosy research is that the mycobacterial resistance is very rare. And in the middle of this slide, you see where M. leprae and tuberculosis splits. So that's about 66 million years ago. There is, however, another mycobacterium called Mycobacterium lepromatosis that split from a common ancestor from M. leprae about 10 million years ago. So still an enormous amount of time ago, but that causes a similar disease, so also leprosy. And so, because there's a lot of overlap in global incidence, and because both of the diseases are poverty-associated diseases, it helps to work on both, especially when developing diagnostic tests. And as you can see here, it's mostly the tropical areas and low and middle income countries where both leprosy and tuberculosis are more endemic. However, in the past, leprosy has not only been studied as an infectious disease, but also actually as a model disease because it has some characteristics in common with autoimmune diseases, for instance. And also, the detection of the pathogen was done first in leprosy. So, M. leprae was the first mycobacterial pathogen that was found to be a causative agent for mycobacterium disease. So before MTB and ulcers were found. Also, but that's maybe a bit too far into immunology. So it was Jon van Rood, a famous transplant surgeon and investigator that detected that for leprosy, there was an association between HLA and disease, which was later also found for other diseases, but so first in leprosy, and also some other findings in immunology that were groundbreaking were done first in leprosy. So it's not only an infectious disease, that's not the only reason why we study it at Leiden, but also because it can be used as a model disease for some other diseases. Okay, a little bit about the background of leprosy, because you may, think, why study leprosy? Is it still a problem? Well, yes, it is, and it has been already so for centuries. And there are still bacterial DNA was found about 2,000 years ago. It has been frequently mentioned in the Bible, but also history books have shown that likely it was that Alexander the Great has transmitted leprosy from the Mediterranean, from the east to the Mediterranean area. And then again, the Crusaders in the Middle Ages, they have been carrying it with them back to Western Europe. However, it has been endemic in Europe in the Middle Ages, but since 1600 is very rare in Europe. And that is also why probably in Dutch textbook for medical students, these are the only pages, the two that you see now, are the only pages with information on leprosy in a microbiology textbook. Of course, we try to make a difference in that, but it is a disease that is in the West forgotten a lot. So a little bit more introduction about the misconceptions that are often around leprosy, and that is what is not very contagious, is not a genetic disease, even though it occurs in families. It's not deadly. It's certainly not untreatable because it can be treated with multi-drug therapy, and of course, it's not eliminated yet, unfortunately. So what is leprosy? It is a poverty-associated disease that is well established, caused by M. leprae or M. lepromatosis, that share a common ancestor with MTB and BCG about 66 million years ago. It ranks second after TB as a severe human mycobacterial disease, affects the skins and the nerves, and causes irreversible handicaps. Well, in Europe, as I said, was not endemic anymore after the Middle Ages. However, there was still one country, and surprisingly, there was Norway, where it was still endemic in the 1800s. And other than that, it was mostly endemic in tropical areas, as you can see from this map. And nowadays, most cases occur in India, Brazil, and Indonesia. And it's striking that there is every year about the same number of new cases, which is about 200,000 globally. And although treatment is very efficient, it needs to be detected early, because otherwise these irreversible handicaps, as I mentioned, will occur. In the Netherlands, my group works closely together with the Department of Dermatology, of Tropical Dermatology in Rotterdam, where Colette van Hees has the expertise for leprosy and sees a lot of patients from the Netherlands. But still, it is an import disease, meaning that there is no active transmission in Europe. So all the cases are import cases. And usually these cases have contracted the infection in their country where they originated or where they were born. And then without any symptoms, they migrated to the Netherlands, and then for some reasons, for instance, older age or use of immunosuppressive drugs, they developed leprosy. Well, since 1945, there have been about 2,500 new leprosy patients in the Netherlands, and we see about 5 new cases every year. And what was striking, and what also has the link to the tropical areas, is that here you see the number of new cases in the Netherlands over time since 1932. And the arrows indicate to the peaks in the new cases, and these coincide with the independence of both Indonesia and of Suriname. So how did they control leprosy in the past? That was very clear. There was seclusion. So in the Middle Ages, outside the city walls, they were kept. And also later, colonies were established, so leprosy colonies. For instance, the former Batavia plantation in Suriname was established as a leprosy colony, but also in Crete, in Europe, in Spinalonga, there was a leprosy colony, and also in the Netherlands, in Heerde, there was a leprosy colony, and most cases that when there were individuals that had contracted the disease in Indonesia. So something more about the bacterium itself. So it is a acid-fast bacterium like MTB, low virulence, as I already said, and that is because there has been a lot of reductive evolution, meaning that the bacterium itself lost a lot of genes during evolution, and these genes are absolutely required for survival. So as a consequence, it does not grow in vitro, and that hampers a lot of the research that we do, because, for instance, for TB, we can culture MTB and we can do studies in mice or in vitro with them, but Mycobacterium leprae does not grow outside a host. So what is done in leprosy research is it's grown in armadillos, so the gordeldieren, and or in nude mice. So these are mice that lack T cells and B cells, which are essential for your protective immune response against mycobacteria. Another striking, unique characteristic of Mycobacterium leprae is that it grows at 35 degrees, which is lower than the normal body temperature, and therefore it occurs mostly in the extremities, so in hands and feet, nose, and and that is also seen in the individuals, and these are the places that are affected most. And finally, compared to other bacteria, it is a very, very slow growing bacterium because it needs 14 days to double, and whereas E. coli does the same in 20 minutes. So it's a bit of a unique bacterium, but this offered also opportunities, because this bacterium, because it grows very slowly, it also mutated very slowly. And this means that over the years and over the centuries, there are mainly 4 strains that are called SNP types that have been detected all over the world. So SNP type 1, 2, 3, and 4, they're called very easily, and that they have a specific a nucleotide at these loci that are indicated on the left. And later on, so this was published in 2005, and later some subtypes were also discovered, so subtypes of 1, 2, 3, and 4, and that offered also a way to follow the distribution of the SNP type over the globe, and thereby making also a migration map, so and transmission map, so to speak. Because as you can see here, some of the SNP types, for instance, SNP type 1, which is mostly present in Asia, and that's indicated in yellow here, and SNP type 4, however, originate in West Africa and is then similar to the SNP type 2 and 3 that are mostly in Europe. They both migrated, and a little bit also on the yellow one, so SNP type 1, they migrated to the Americas. And in Americas, you see all of them. So this led to the following migration pattern. So the origin of leprae, Mycobacterium leprae, was probably in Africa, and so in Ethiopia, and then it migrated to the east and had a special SNP type there, and then also using the Silk Road back to Europe, and then with slave trade and with the colonists back to the towards the Americas. As I told you, leprosy, Mycobacterium leprae grows very slowly, but also the incubation time can be very, very long. So incubation time, so it's the time between infection and the colonization of the bacterium in the body using well, a lot of mechanisms that I will not go into, but it can stay undetected, not causing disease for many, many years until like immune suppression or old age occurs. And this is depicted in these slides with an example of a chimpanzee from Sierra Leone, was captured there at a young age and then brought to Japan in the zoo. In Sierra Leone, leprosy is endemic, and in Japan it's not. So it's unlikely that it contracted the infection in Japan. But then still, there were about 30 years almost that Mycobacterium leprae was present in this chimp without any disease being averted. This is also recently published for human leprosy. Some people called it, well, they probably had the infection for many, many years, but developed clinical signs only after 50 years. And of course, some circumstances can affect the occurrence of leprosy, and we see that also that climate changes in human and animal environment, or in human animal interactions, and also the habitat of animals that is available for them, that really aggravates the adverse effects of Mycobacterium leprae in the environment. And one example for this is leprosy in the USA. So leprosy is not endemic in the USA, but around 2010, what was published is the following, is that there were many cases in the southern states of the US, as you see depicted here on the left and the map. And these states also are the habitat of armadillos, so the animal which Mycobacterium leprae can also be grown. And what was used was these SNP types present in infected armadillos, as well as in humans living in the same areas. And they compared that, and then they saw that these SNP types were similar. So this indicates that there was a likely zoonosis in these cases, in these human cases, because those individuals did not travel to any tropical areas, so they only stayed in the US, but they likely had contact with armadillos. So this is, for instance, a picture in which you can see how such contacts may have occurred. So it could, of course, be hunters, because several hunters in the southern states of the United States hunt on armadillos for barbecue meat or for making all kind of baskets, souvenirs from it. But also people that have never traveled to endemic leprosy endemic areas. For instance, this case report of a Canadian man who did travel every year to Florida. And so as depicted here, because some of these animals, well, they are in the gardens or they are in swimming pools. So it could be that Mycobacterium leprae present in these animals was transmitted to humans that way. And it even got to the New York Times last year, whether or not maybe leprosy is endemic in Florida. And currently we're doing research together with the University of Florida in Gainesville to see whether individuals without any symptoms may be infected, and to do a serosurvey in that area. And what we also did is, and that was together with the National Hansen Disease Program in Baton Rouge in Louisiana. They have a armadillo model for Mycobacterium leprae, and they sent us some samples, and we used our diagnostic test, which is a very easy rapid test for detection of infection, and we showed that we could also use our test for detection of leprae infection in armadillos. And this is now also something that we're going to do in Florida, where these wild armadillos are caught, just to see how far advanced this infection in these animals are. Some of the studies are also taking place in French Guinea also. And then something that surprised many of us in 2016, it was published in Science. And what was shown here is that there have been some sightings of red squirrels in the northern UK, so in Scotland, or on an island, the Ramsey Island in the southern UK. And there were no human cases around it. And actually, for centuries, there have been no human leprosy cases in these areas. So what they also did, based on the sequence of the bacterium, based on the SNP types, they identified what SNP type was present in these animals, and they discovered that it was mostly in Scotland. It was mostly M. lepromatosis, which is in humans not that common, and in southern UK, so on the Ramsey Island, it was Mycobacterium leprae, and the Mycobacterium leprae strain that was identified there, that was also very, very similar to a Mycobacterium leprae strain that was detected in skeletons of medieval graveyards of leprosy patients. So this means that in those, well, hundreds of years in between, no human cases have been present there. The bacteria, because I told you that the bacteria needs a living host, has to have been somewhere. So even, I mean, it could be in these red squirrels or in a smaller animal. We still do not know. And so therefore we started collaboration with the scientists from Roslin Institute in Edinburgh, in the University of Edinburgh, and in which we did not use roadkill animals, as was done in the science paper in 2016, but trapped animals, and we followed them in time. And what we saw is that when there were no clinical signs, our test was also negative, but when there were clinical signs, our test was positive, and when more clinical signs developed, our test became positive. So again, indicating that we can use this test for infection that we also use in humans, and that is a rapid test, so that we can also test that for detection of infection in animals. Of course, we also tried to find lepre or leptomatosis in red squirrels in the Netherlands and in Belgium and in Luxembourg, but all of the samples that we got from RVM in the Netherlands, from Belgium and the Netherlands, they were all negative. So they did not contain any DNA from Mycobacterium leprae and Mycobacterium leptomatosis. A similar study was done by other researchers in France, which also did not find any animals. So it seemed to be exclusive for the UK. Okay, so finally, I want to show you also some of the research that we did in soil, because we're looking for another host besides the known animals and besides human. We thought we may discover whether some places Mycobacterium leprae can survive for a while in soil, or maybe in amoebae that it can survive there. So we have a long-standing collaboration with Bangladesh. So we collected samples there in the houses of leprosy patients. We also collected samples in Suriname. There were around in the armadillo burrows in, for instance, the Batavia plantation, and on the British Isles in the neighborhood of the red squirrels. And so we only had a few samples that were positive, and only of the samples in Suriname and Bangladesh we could type. We had enough DNA to also type the samples, and they were derived from genotype 1. And some of you may remember that's the genotype that occurs in Asia. And so this indicated that, well, for Bangladesh, it's to be expected that it's type 1. It also occurs in most of the patients there. For Suriname, it could have been type 1, but it also could have been type 4. So it went with migration of the people colonizing the Americas. And this was informative to know, but of course it was only a few samples. So then we also collaborated with Jaap van Dissel in his search for leprosy in Perke-Donders. And what was interesting in that study is that not only did he do excavation of the skeleton of Perke-Donders, who was a priest that took care of leprosy patients in Suriname, but also of a leprosy patient. And the leprosy patients we found to be very clearly genotype 4. And genotype 4 is the genotype coming from can't show it now, but it comes from the slave trade, as well as from the European colonists. So it was interesting because it's different from the other side. So in Suriname, it is like it is, for instance, also in Brazil. It was interesting to see that we find different strains, so indicating the migration with humans of the Mycobacterium leprae. And then finally, of course, we don't know, as I already said, where Mycobacterium leprae and leptomatosis had been during all those centuries in which no human leprosy cases were detected in the UK. But also for instance, the armadillos, we know they carry Mycobacterium leprae, and a lot of them are infected, and it shouldn't be a problem if you don't come into contact, and if they have enough area to live in. And but if like is ongoing now in Florida, I have a lot of golf areas or new houses are built, then these animals will come in contact with humans, and we can contract leprosy from them. But originally, there was no leprosy actually in the Americas. So we brought it there, humans brought it there. And so now these animals are giving it back to us. So I would like to conclude with this. So this is currently still very interesting. So the interaction between animals environment and humans that surrounds leprosy, but also still leprosy in Suriname is some of the topics that we are interested as a research group and would very much like to collaborate on. So thank you very much for your attention. And if there are any questions, please let me know.
Thank you very much, Annemieke. Very interesting talk. Are there any questions? So please raise your hands and then we can see who will be on top of the list. And before I see anyone, I can ask a question myself, because so first of all, you said that leprosy was very difficult in transmission, but in the end, you reported a lot of apparent transmissions across species, between humans to other animals, maybe also among animals. What is necessary? Is it when we eat animals or touch animals or blood contact? Is that what?
Well, for the
necessary?
armadillos, yeah. For the armadillos, for instance, they have a lower body temperature. So unlike humans, if they carry Mycobacterium leprae, they carry it throughout their blood, throughout the system, and whereas humans carry it only maybe in their extremities. So if you have a armadillo and you capture an armadillo and you're dependent for your protein diet on such animals, like some people in Brazil are, it lives in your household for several days until it's slaughtered. And then, of course, some of the people like to eat it as ceviche, so raw. And we have shown that there's an association with eating raw armadillo meat is dangerous. I mean, it is still eaten. Some people who know it can transmit Mycobacterium leprae, they also still eat it. So but I wouldn't say it can be transmitted easily. It depends, of course, on whom it's transmitted to. Because if your immune system is optimal, then you're able to kill the bacterium. But if you get large doses, I mean, of your immune system is not able to kill Mycobacterium leprae directly, then you may develop it over the years. Yeah, but it's not it's not like with Mycobacterium tuberculosis. If you're on a bus in South Africa and somebody coughs in your face, then you can contract it directly.
Yeah, and because the relationship with eating animals makes it also unlikely that, for example, rats or mice in the house, or even cats and dogs as pets, that they will be a vector.
Yeah, we don't know. I think they may be, but then they need to shed it. So I think for instance, the person contracted leprosy by going every year to Florida. He didn't interact with armadillos, but the armadillos are there. So infected armadillos are there. So it could be shed by these animals. If rats are able to carry it, maybe the same. Yeah.
Nicolas, you have your hands up. That's because you want to start your talk, or do you have a question?
No, because I have a question. Thank you for the presentation. No, the story of Father Peter Donders, I wanted to ask. Can you explain that again? Because I missed.
Okay. Yeah. So Father Donders was a Catholic priest who lived in Suriname, and he cared for leprosy patients. And the Catholic Church so wanted to investigate whether he has had leprosy or not. And so that was the idea. So what they did is an excavation, and the skeletons usually in some part of the bones or some part of the teeth, then Mycobacterium leprae can be well preserved. This is also how we identified Mycobacterium leprae in skeleton of a medieval leprosy patients, for instance. And but it was negative for Peter Donders. So this meant that no Mycobacterium leprae DNA was detective.
I always thought the story was that he had leprosy before he died. But
No, well,
and that's true.
if he had leprosy, there were no signs on the skeleton that he had leprosy, because for some patients that is really visible. Nor were there any leprate DNA. Was there any leprate DNA found?
Okay.
Okay. Thank you very much, Annemieke, and thank you.
Yeah, you're welcome.